Friends, 25th April is celebrated as DNA Day. Speciality of this day is with the two main factors or facts. One is the discovery of double helix model in 1953 and the day of completion of Human Genome Project in 2003. The three-dimensional structure was first identified by Francis Crick and James Watson at the Cavendish Laboratory of Cambridge University on 25th April 1953, whose model building efforts were guided by different scientists' discovery. The basic model was drawn by Crick's wife Odile. The basis for three-dimensional structure was the contribution of different scientists. First and foremost, Swiss scientist Friedrich Mischer in 1869, who coined the term nuclein, but the problem was he could not identify that particular component as DNA. The subsequent discoveries like Griffith experiment on transformation in 1928 on Streptococcus pneumoniae, subsequently 1944 Avery McLeod McCarty's experiment on the same transformation principle by using the same organism Streptococcus pneumonia. He found that something is genetic material, but he could not say 100% it as the DNA. The third scientist, or we can say the fourth scientist who contributed in this way was Hershey and Chase. In 1952, they found DNA as the genetic material who worked with the bacteriophage. This is the basic principle or basic scientists who worked for the DNA discovery, but for the three-dimensional model or the helix structure of DNA was contributed by four important scientists. First one was Rosalind Franklin, who is called as Dark Lady of Genetics. She was working with X-ray diffraction pattern, which was obtained by the postgraduate student of Raymond Gosling. Means Watson and Crick obtained this particular information from the postgraduate student Raymond Gosling. Wilkins, another scientist, also worked on X-ray diffraction model. He proposed helical corkscrew-like shape of DNA based on X-ray diffraction pattern along with Franklin. The third scientist was the organic chemist named Alexander Todd, who determined that the backbone of DNA molecule contained repeated phosphate and deoxyribose sugar groups. The fourth scientist is Chargaff, who found that amount of DNA and its four bases as ATGC. Before Watson and Crick, Pauling proposed the double helix model of DNA, but it was unrelated. On 25th April, in Nature journal, this particular information of three-dimensional model of DNA was published. The major fact or the unknown fact about this double helix model is these people never worked on this particular model. Means they were not the basic workers about this model. Watson and Crick collected the metadata on the basis of above said four scientists, namely Franklin, Wilkins, then Todd and Chargaff, and they just concluded that three-dimensional model. Means they just drew and they made a particular article and sent to the Nature journal, and it was accepted worldwide, and it was the peak period in 1953 to accept it as a universal model. Watson and Crick developed their idea about genetic replications in the second article in Nature published on May 30th, 1953. For their work in 1962, Watson, Crick and Wilkins awarded Nobel Prize in the field of molecular genetics and biochemistry. Let us talk about DNA. It's a molecule composed of a chain that coil around each other to form a double helix carrying the genetic instructions used in the growth, development, functioning, and replication of all the known organisms and many viruses. DNA, RNA, and proteins are the known genetic material. In the central dogma, we can find the information flows from DNA to RNA, then to protein. DNA cannot be only in the double helix, it may also be in single helix form. The single-stranded DNA is found in Gemini virus, which is a major group of plant-infecting viruses. Double-stranded DNA is found in almost all the higher eukaryotic organisms. The two strands are also known as polynucleotides, which are composed of single or simpler monomeric units called nucleotide. The nucleotide is composed of nitrogen containing nucleobases, that is adenine, guanine, thymine, cytosine, called deoxyribose, and a phosphate group. Nucleotides are joined each other by a covalent bond between the sugar of one nucleotide and phosphates of the next, resulting in an altering sugar phosphate backbone. The nitrogenous bases of the two separate polynucleotide strands are bound together according to base pairing rule. The complementary nitrogenous bases are divided into two groups as purines and pyrimidines. As you know, adenine and guanine comes under purines and thymine and cytosine comes under pyrimidine. If it is RNA, means in place of thymine, uracil will come. Coming to DNA, here both the strands of DNA store the same biological information. This information is replicated as and when the two strands replicate separately. Large part of the DNA is non-coding, meaning that these sections do not serve as patterns for protein sequences. The two strands are antiparallel or opposite to each other. These are the basic units in central dogma where the genetic information flows. Coming to the facts about DNA. DNA is deoxyribose nucleic acid. It is found in all the living organisms. It helps in identifying the parentage and the criminals in any incidents. It helps to identify evolutionary pattern. It gives the rareness of organisms. DNA can be obtained from very common material like hair, nails, skin, blood stains, tissue, leaf samples, seeds, anything. Mutations in DNA may lead to risk of diseases like dangerous diseases like cancer. Change in one DNA base also leads to blunder. Many things cause mutation, including UV rays, and that cause major damage to DNA or major changes in the DNA. DNA acts as a hereditary process. The mitochondrial DNA, where eggs are found, this passes only from mother to child. All cells in our body contain DNA. Exception is red blood cells. If we start typing the human genome information, it takes about 50 years. Every human shares 99% of the DNA, but the identical progeny or the progeny of a single parent shares 99.5% of the genetic information or DNA. Though mitochondrial metabolism it generates energy for the body. Identical twins share about 100% of their genes in case of human or animals. But in plants, the progenies are acquired. If it is acquired from homozygous and homogeneous parents also, the offspring differ because of the environmental impact and the major mutational changes. The process of genome sequencing was first put forth by Frederick Sanger, and his discovery or his invention was the major basis for Human Genome Project. This became a basis for several genome projects of other organisms, maybe rice, Arabidopsis, many of the plants, and higher organisms, even the lower organisms. Now the different generations of sequencing, like now it is third or fourth generation, which enabled the very little cost or reduced cost for genomic sequencing, and the process of sequencing is extending to almost all the organisms which are existing on the earth. The resequencing process helps in identifying the rare alleles and utilizing them in the major leading or we can say improvement in different plants or animals, or transferring the genes for particular traits like resistance for biotic or abiotic stress. Friends, 25th April is celebrated to remember the discovery of different scientists, to thank their contribution and boost the young researchers to get motivated.